So there's many reasons to do biological research, but one of them obviously is because what we would like to do is fix the human body when there are diseases. And so if we imagine ourselves as people fixing equipment, we reach down into the human body and we look inside, and this is more or less what it looks like. It has a bunch of parts, and the only problem is that for half of the parts we have no clue what they're doing. So for instance, this piece here, I don't know about you, if anyone knows what this is doing, I certainly have no idea. And the problem is that if this equipment is broken and we don't know what half of the pieces are doing, the chances of being able to fix it are almost zero. And that's what's happening when we look at human biology or animal biology, that roughly half of the genome is unannotated. No surprise that we have no cure for most types of cancer, for diabetes, for neurological disease, and so forth. So what do we need to do? We have to understand what the other half of the parts are doing. We can't start taking apart humans, so the idea is we investigate an evolutionarily related organism. And one of these organisms, for instance, is Drosophila, but we can also work with worms, fish, yeast, and so forth. And the idea is that since we're related and descending from a common ancestor, the biology is conserved, and what we learn in model organisms then is also to a large extent true in humans. And so even though we know this conceptually, I've heard many times people thinking that this is not the case, just like twins who obviously are very related genetically, but when they become teenagers, they might think that they're totally different from each other, but they're not. And the same is true here. So I just want to give you one example of this, and that's illustrated here. So a few years ago, we discovered a transcription factor, a protein that controls gene expression, and this transcription factor, when it's mutated, makes flies very lean. This is a quantification of the total fat in the body of a control fly, and you can see here that mutants for this factor called Reptor are very lean. They have less than half of the amount of triglycerides. This is true in both females and males. And this is essentially the strongest metabolic phenotype that we've ever seen in the fly. Now this work was then used a year later by another group that was studying obesity in the Samoan population. And that's a great population to do genome-wide association studies because of its homogeneity and the prevalence of obesity. And they were able to narrow down a specific region in the human genome that strongly affects adiposity. But in that region there were still many genes. And when they then saw our work, they realized that one of the genes in that region is the human equivalent, the human ortholog of Reptor, and it's called CREBRF. And in the end, that led them to discover that this genetic variant in CREBRF has the largest effect on human adiposity that has ever been discovered. So they found here an effect size of this SNP that is much larger than that of any other known body mass index risk variant in the human population. So essentially what we have here is that the gene in the fly that has the strongest effect on adiposity in the fly that we've ever seen is then equivalent to the gene in humans that also has a strong effect in human adiposity. And so now that we have tremendous sequencing technology, that we're able to read the genome of humans and of even disease states and cancers, the problem still is that we don't understand half of what we're reading. And the information from these model organisms gives us what we need to interpret what we read in the human genome. And so my plea and my thought is that the more we want to cure human disease, the more we need to study worms, flies, yeast, fish, and so forth. So thank you for your attention.